Let's get started. So everybody, welcome to day 2 of workshops. This is the second workshop of the day, but this one is the later one at 1 p.m. So let's get started. So my name is Vera Estrada. I am from the MIT Media Lab, part of David's group. I've been helping organize the Bio Summit for the last 3 years while also doing research. So it's been a fun time. But jumping right into things, if you haven't changed your naming convention, do so right now. Your name, your pronouns, your affiliation, and the country where you're from. Just to start, people may mute themselves. Participants do have the ability to unmute, ask questions. I think this is an interaction-based workshop. We want people to comment, make questions, so feel free to speak up. And if not, you can drop questions and comments into the chat, and we'll try to be receptive to them as much as possible. To start, we would love if you kept your video on. We would love to see all your wonderful faces as we went through this workshop. All right, continuing. If you haven't checked out our website or our Slack, do so. There are the links. All the materials and notes from today will be in the participant Google Drive, the third link. The material from yesterday is being uploaded or has been uploaded already. Today's material will be uploaded at the end of the day here on the East Coast. And if you need to contact the Code of Conduct team for any reason, please email them at codeofconduct@biosummit.org. And of course, we must give a special shout out to our sponsors. Without them, we wouldn't be here. Yes, the Bio Summit is virtual, it's not in person, but we still need them to make this happen. So we're very grateful that they support us each and every year. And without further ado, I'm gonna hand it over to Roland, who's handing it over to Marissa, who will be our MC for today and will guide us through the second iteration of guided tour of the biocreation stations. So Marissa, please take it away.
Thank you for the introduction. Let me figure out how to share my screen properly. One moment. Make this full. Is this coming up as a full screen? No, it's not. Okay, now it is. Okay, so first off, I'd like to just shout out the amazing people who put together the biocreation station. And that includes Carolina and Heidi and Alexandra, who helped put together the welcome kit. So in looking through the gallery, there are so many wonderful works that we could spend hours speaking about. I picked 2 artists to speak about today, and if there's time, we can open up the conversation based off of what caught other people's eye. So I want to begin by placing Corinne Akata Takara's work in conversation with Alexandra Janis's work. A common theme that has been popping up this weekend is this movement between macro and micro needs of our communities. And within Corinne's work, I see a focus on the relation between the personal, memory, family, materiality, and the spaces that exist between these things. And within Alexandra's work, there's a highlighting of the opposite, kind of the combustive nature of artificial flavoring, how this synthetic process is unknown to most of us, and what brings us our contemporary flavors of today's food, pointing to a pretty dystopic reality in which the human animal is connected through multiple iterations of what was once something that grew from the ground. And so with that brief summary, I'm going to introduce Corinne's work. So this is the Gilded Fungal Flowers Bio Quilt. Here it is, and what stood out to me with this work is the incorporation of materials that were utilized in the cooking process and growing up, and that's speaking to culture and family, and how all of these are intertwined within personal and collective memory. So Corinne, if you would like to speak to your work, because you can speak to it better than I can, you're more than welcome to.
Thank you. Thank you so much for that introduction, Marissa. So yes, this is a piece of work that I developed recently. It's actually a prototype for a larger project in which I'm going to be inviting communities in San Jose, California, 3 very siloed communities, to make their own quilts. So I thought that the metaphor, the idea of making quilt tiles would be a really interesting way to engage communities in these unfamiliar conversations about biodesign and bioart through a medium or through a metaphor that was accessible. And especially since we're still in time of COVID and we're remote, what I'm going to be doing is sending out through community gardens some of the ingredients from their own neighborhood so they can grow their own mycelial structures on a quilt tile section and grow from their own neighborhood materials, cultural patterns and incorporate their thoughts. So this piece here was the start of that. And in it, I have mycelium that grew in substrates. All the materials here are locally sourced. So the mycelium was grown in coffee bean husks from Dandelion Chocolate Factory in San Francisco. So that's within like a 55-mile radius, not too far from where I am in Cupertino. And as well as use coffee grounds. The little tiny flower pieces are chitin bioplastic. So I am half Japanese, my family's from Hawaii, and we have a lot of history of actually eating a lot of the materials here. So chitin is in the shells of shellfish. And growing up, we actually ate the shells. So it was part of our diet. You don't peel something and throw it away. You actually, generally we would fry these shelled creatures, usually from the valley streams of Eel Valley, and fry them with hot chili pepper, and you eat the shells, the chitin shells. The bits of little green constellations knotted, that's kombu seaweed that we use in our soups. So it's kind of the base for our soup stocks. And the way you prep it for a soup is you soak the seaweed in water, and then you tie it in knots, and then you cut between the knots so that you have these shorter lengths. But it's kind of a communal family thing to do. So as you're tying the knots, you're talking story. In Hawaii, it's called talk story. And so I wanted to include that in here because a lot of the other biomaterials, bioplastics I explore use agar-agar. So agar-agar, I think a lot of scientists think of that as what you plate your culture cells with. But it was discovered and invented essence into use by the Japanese, and then it propagated throughout Asia as a dessert ingredient, like, you know, the jello part of a dessert. So seaweed, I think, is really important in our conversations about the future of sustainable design. And I wanted to reference that as both to my family culture with the seaweed knots, the kombu knots, but also that seaweed is used for bioplastics. The molds I used for these were designed in paper. So I laser cut paper structures that represented Japanese flowers that I have on one of my kimonos. And so I did just one unit, and I laser cut it and dipped it in hot wax. And that became the mold in which I stuffed the incubated bark, the mycelium incubated bark, Ganoderma mushroom incubated bark, and the coffee bean husks and the chocolate husks. I don't want to go over time, so I'm just going to stop in case I'm going over.
Oh, no, you're fine. Yeah. I figured you could speak a bit about your work, and then Alexandra could speak a bit about hers, and then we could have, maybe if people are interested, a discussion about of these two works that have to do with food, right? But on very different levels, utilizing concept of food and flavor and things like that.
And I just want to thank Daniel Ruskin, who's here, my goodness, for putting in my article link there. But I think at the heart of it, biodesign is communion and conversation. And so most of the art projects that I do, or the bioart projects, they're all designed to be in conversation with other people. So let people have their own experience of feeling the materials and growing something and be in conversation with storytelling in the past and future and the present. It's a multi-generational experience, I think it should be. So for me, I'm not a scientist. My training is really as an artist, and my experience with biomaking is through family. So my dad's from Maui, Hawaii, and I moved about 11 times growing up, but every summer we go back to Maui. And my experience with biomaking is that you really have to know the names of the organisms, whether they're plants or animals, know the indigenous names, know in my case, it was Hawaiian names and the Japanese names, and know the stories that go with them, because every material, every organism has a story of how it fits into place. So that's kind of how I frame my work. And I'm hoping to engage other communities in my area here to have cross conversations through these bio-grown quilts.
Yeah, that's very beautiful. Okay, maybe we could pause on that and just move over to Alexandria's work. And here we are. I don't know if this is, okay, yeah, I just love, first of all, visually, these are just very beautiful objects without having any words attached. Let's see, okay, we'll start here. So based off of the gallery, Alexandra is a speculative designer, a UX designer, and a bio artist, which I feel like we all, maybe not all of us in this talk are, but I feel like all relate to that. So this project, Atuma, is a collection of spices which creates flavor through pure volatile organic compounds, inspiring people to combine, explore, and experiment with different flavoring combinations, which is a really interesting idea to me in consideration of how kind of closed off food science or flavoring, synthetic flavoring can be. So I'm very curious to know what your experience in working on this project is. So let me look for you. Let's see. I saw you a minute ago.
Actually, hi.
Oh,
I can just unmute myself.
Oh, okay, perfect.
Hi, thank you so much for this introduction, Marissa. I think it's just so cool that that I can be part of this and that we are doing this over here just to share work. Yeah, thank you so much for doing this tour. Okay, yeah, so as you already mentioned, so this is about flavor, and it's a speculative work. And basically what you see there is a series of spices that I made from the 24 major volatile organic compounds of strawberries. So when you think of flavor, not as like coffee flavor or apple flavor or strawberry flavor, but in the molecular sense, flavor is a combination of molecules, and which we mainly also perceive through the nose. So they are olfactory, and there is a term called retronasal smell. So we smell the food that we eat from inside of our mouth, and then we think this is like taste, but actually it's smell. And this smell is made of molecules, and in total there are like 11,000 molecules that we can perceive as flavor in our mouth. And so the flavor of coffee or of apples or of strawberries is just a combination of molecules. And yeah, so each of these molecules has a flavor of their own. So just because a molecule is inside of strawberry, it doesn't mean that it tastes like strawberry, but it's all about the amount of it and the mixture together with other molecules. So one molecule, for example, furanio, is a caramel type molecule, and it's found in strawberries. but also in coffee or in bread. So yeah, each molecule with a specific flavor gives like creates the flavor profiles of different foods. And when I researched this topic and I got to know molecules, then I thought, wow, this is such a crazy tool for humans to create a maybe more sustainable versions of current flavors. So if you imagine like, so when I started this, before I started this project, I ate a strawberry yogurt, and the packaging, there was a description that this yogurt doesn't contain any artificial flavoring, and I thought, oh, well, but it's a little bit crazy to use up all the resources like water and soil to create strawberries to then put them in yogurt. And then if you imagine if everything was flavored naturally in the supermarket, like all the consumer products like chips or hamburgers, then is this really a sustainable way, or can we actually use the science and like create synthetic flavors to produce something that is more sustainable? But also, so this is like one thing that fascinated me, but also the potential to create flavors that didn't exist before. And actually, then I found out there is a whole industry that has been doing this for the last 50 years. And it's a massive, really, it's like a multi-billion-dollar industry, flavor and fragrance. And I was in touch with some of the major companies. There are not so many companies that are doing this, but they have a lot of power over the market. So if you imagine a supermarket, all the foods, like a flavorist could go through the supermarket and just like point out the foods that they help create, like the flavor profiles of the foods that they made. And yeah, this gives industry a lot of power to make us dependent on their products, and to create flavors that are addictive, to create flavors, good flavors for products that are not good for us. So I wanted to democratize it, and democratize flavor molecules so that people could actually do it themselves. So I 3D printed 24 strawberry molecules, and I color coded them according to their flavor profile. And so in theory, so they are made of cocoa butter, and because molecules are mostly fat soluble, so they are supposed to be flavored, or I flavored some of them with like each single molecule that they represent. But then in the process, so I had my 24 molecules of the strawberry, but in the process I found out some of them are explosive, so I couldn't do that. Like I couldn't flavor that, but some I got. And it's also really hard to order volatile organic compounds as a design student from a company like Sigma Aldrich. They will not sell that to individuals. I mean, it's also dangerous. Also, many are toxic. So, and also when I gave people spices to try it out in food, actually they didn't like the flavor of each molecule, which I think is funny.
What did they not like about it? It was
the taste is like really flat, because each molecule is like inside of a food, but it's like a building brick of flavor. So it reminds you of something, like maybe it reminds you of pineapple or banana, but there is not enough complexity to the taste, which is really important. So for example, if you eat like maybe a really bad chewing gum and you think, oh, this tastes like raspberry, but like really bad, I think this might be because there is not enough, like there is maybe only 2 molecules in there, like maybe raspberry ketone and something else, but it's flat. So when I gave molecules to try for people, so yeah, and maybe I should mention, so these are supposed to be rasped over food, like in different combinations, like Parmesan or ginger. But yeah, so I think humans are not like we struggle with, because our chemical sensors, they evolved to protect us from putting stuff into our mouth that is dangerous. So if we taste something we don't know, then we don't really, like, we should be mistrustful first, right? So I think it's quite difficult for people to accept things like, yeah, it's, I'm fascinated by it, because when we have visual art, like it's easy to look at something that you don't like, but it's much harder to taste something you don't like, because it could potentially be dangerous for your health. So that was really interesting. And at the moment I'm wondering, because this was like a proposal, and it's like an ongoing process of like having molecule tastings sometimes, and like thinking about how to make this tool of chemistry, of food chemistry, more like how to actually empower people with it, how to make it actually, how to create a usability for it, like a good UX experience of flavor molecules, maybe. Yeah, so this is, yeah, I think this is it from me right now.
There are a couple questions in the chat. I can
yeah.
read them. Corinne asked, did you 3D print the molds and then fill them to create the forms, or how did you make those?
Yeah, so basically what I did is there is a website, it's called mold.org and .org, and so if you put in a chemistry formula there, then it gives you a spherical model, and then based on the spherical model of each molecule, I made a 3D model on, I don't know, Fusion, and then I printed it, and then I casted it. No, I made a silicon mold, and then I casted them from my plant fat material. Yeah. And then in the process, like you add the molecule into the fat. Yeah.
Awesome, thank you. And then Aliki, I'm hoping I'm pronouncing that even remotely close, asked, do the colors of the food play any role in the taste?
Okay, now I'm thinking, like, in general, do colors play a role, or in case of, or like why I gave a specific color to each of those. But yeah, maybe I can say, maybe, one second, maybe Aliki, can you specify?
Sure. No, I was mostly thinking if there is any difference in the molecular structure according to the colors of the food. So I have been experimenting with like only green food, only red food, or only purple food. Do they make any difference in the molecular level?
You mean like, you mean not specifically the project, but just in general, the how
what
how
other aspects of food make it different? Like what distinguish them also? Do the colors also, like tastes, create the difference in the experience, or have you seen any difference in the molecular according to color? If you have searched for it, of course.
Okay, so like like in general with food, I would say definitely there is some, like, color is an indicator of specific flavors. So or like we have evolved with plants in a way that they signify through, like red, for example, is sweet. We have evolved to with plants in like the symbiotic relationship to experience the color red as something that is indicating either sweetness or maybe, yeah, also blood. But like when we see an apple, a fruit is ripe when it's red or bright colored. And when something is green, then this is like more an indicator of toxins like alkaloids, or yeah, like that something is bitter. Bitterness is associated with green. And brown is, for example, actually a processing indicator, like a visual indicator for caramelization, Maillard reaction, like everything that's becoming brown is like roasted. It's like it's a later development of our involvement with like the visual appearance of food. But I want to say that in this specific case of the strawberry flavor, so it's 24 molecules, but as I said, they don't all taste like strawberry. Like actually none of them taste like strawberry. And so the colors that you, for example, see in this picture, I use them like because all molecules are grouped according to flavor profiles. So for example, there is one here that is light green, which is I used as a color to indicate grassy, like grassy flavor types. Then in the background, there is a light yellow one, which is cheesy. Yes, there is the strawberry shares a flavor molecule with gorgonzola cheese. That's why they fit so well together. And then there is also moldy molecule in the front, which is light purple, and there is a caramelly one, which is furanol that I touched on before. And there's a red one, which is fruity, and a dark purple one, which is ethereal. But I took these, like, it's not my, I took this information from the flavoring industry, because there is a website, it's called goodsense.com, and they have a data bank of all the flavor molecules, and also like according also to groups of where to find this molecule, which like in food, but also in general, how, like which companies sell it, and also which smelling profile it has, like how many nanograms of it you need to have an experience, from which point it is toxic, and like all these things. They have a database for this, which I used extensively also to create the spices.
Yeah, it's really fascinating to kind of think about the differences in approaches and consideration when in your project, Alexandria, with using these very particular, to the molecule in these particular amounts, and having to have an awareness of what might be too dangerous to even work with on an individual, non-institutional level. And in Corinne's project, which is, I mean, everything is about community in some sense, right? But Corinne's project is very different, right? It's like using food and using these byproducts and things that people might not even think about eating, but that is totally an edible ingredient in a lot of cultures, and weaving that in to the work to create this new material. So it's interesting how within biotechnology, it's like we're using these very old ingredients and repurposing them, as well as extracting flavor profiles from what already exists, and creating these, in your case, creating this project that is speaking to these like very minute but apparent distinctions when it comes down to the molecular form, but also, of course, like no one wants to just take a bite out of this form. I don't think that's necessarily your intention. It's funny because to me it looks kind of like one of these bath bombs or something. Actually, when you said cocoa butter, it's like, oh yeah, like that could be a nice like skin treatment. Yeah, it's interesting approach on like working with senses and what is in your surroundings in terms of like scent and like personal history. I wonder if anyone, I haven't been looking at the chat, but I wonder if anyone has any comments or questions as it relates to utilizing these ingredients or food within biotech as it relates to community. Maybe that's too specific, and maybe people just want to talk about what they're thinking about too.
There was a question asking Corinne to talk about her work with the Quilt Museum from Daniel.
Oh, yes.
And about Alexandria background, that's another question that I spotted.
Did you want me to go or address that? First one, or? Okay, just real quick. Okay, thanks. So yes, as I mentioned earlier, this is the start of a conversation I hope to have with community. I think everything is storytelling. I love how Alexandra's storytelling. What is flavor? What is modern flavor? Something we don't think about in a really very interesting way. But what I'm trying to do is hyper-local conversations with community, and I'm collaborating with the San Jose Museum of Quilts and Textiles on a 3-month journey with 3 different communities in San Jose that are pretty siloed. So Japantown, Little Saigon, and East San Jose, Mayfair community, which is Latinx, and inviting each group to explore the materials of their community, their waste products, their local edible materials. For example, there's nopales cactus in this, as well as a cactus fruit bacteriocellulose, the kind of red part of each of those flowers. So I think what my project is aiming to do is to bring local communities into the conversation about innovation, material, sustainability, design. So how do we bring those material conversations, but elevate people's existing knowledge and expertise in food and in their own, just to bring their own storytelling to these conversations, bring in people who are excluded, and a lot of what is innovation. So I'm in Silicon Valley, but there's a huge demographic of people who are not part of the conversation, of feeling they are in either the Silicon Valley conversation or the biotech conversation, which is, you know, going to be the next huge driver in this region, and so many people are excluded from that. So how can we build these conversations from within community, through art, through food, through biology, so that there's ownership and more innovation coming from the knowledge that's already latent and existing within community and culture. And so I'm really excited to a quilt museum, because it's going to result in 3 different quilts that we're going to have at the museum, but it's going to be driven a lot by in-place community engagement, as well as online engagement.
Are there dates for that exhibit?
It's a residency, so it's going to start in January and go through March, and then we haven't set a date yet. And the resulting pieces will probably go out into the communities in different ways. It might be broken up into, the quilts might be broken up and put into the soil at the community gardens. We don't know yet. We're still trying to figure out, like, we need to have this conversation. It's going to grow with the community as to what they want to do with it and what it will be.
Yeah.
Thanks for the question.
That was really well said, Corinne. Thank you for talking about people who don't have access or don't feel like they have access to these platforms. And I think it's really interesting. I mean, we are in the gallery, but I think it's really great and interesting that both you and Alexandra are not trained scientists, that you have artist and designer backgrounds. And I mean, perhaps like that's what this entire conference and organization is about, linking people together who are very passionate about the intersection of biotech, art, and design in order to I don't know how to make the world a better place, have us understand each other more, and art is so important, so much a part of that. And I think it's really great that we can all sit around our screens and be able to really get into someone's work, because often with the visual, especially now, we just see the image on the screen and we either like it or we're not sure. But this kind of conversation is where, you know, it really enriches our experience. Are there any other thoughts that people are having.
I'll rest see more of the quilts if we have more pictures.
Go. Corinne, are there more on your website?
There should have been more in this slide deck at the bottom. You can, but yeah, I can put in my. Isn't there a slide section? I guess not. Oh, there's a little arrow there on the right. Do you see that little tiny gray arrow next to the tiny arrow up a little further to the right? Just keep there and go.
Okay. So sorry. And the panel of people talking was blocking.
Was in the way, yeah.
Yeah.
Okay, part of the process. I'm really interested in grown assembly. So part of this conversation too is how do we think differently about fabrication design so the materials grew into this mesh. So we have the opportunity for, through art and engineering, to explore different pathways in engineering. And then this is laser cut bacterial cellulose. So I like to use a range of tools. While I grew up sitting on my grandma's porch using just hand tools, I now use a lot of technology tools to explore the kind of work that I want to do with communities. So those are just some examples. And then, yeah, I have a lot on my website. I'll throw that in the link, but into the chat. So.
Great. And then I saw someone's hand was raised. Daniel?
Hi there. I just wanted to ask Alexandra a quick question. Can you talk a little bit about your background, what brought you to this project, and what brought you to taste and flavors?
Yes, thank you. It's actually, it's a good question. So basically I was studying at Design Academy in Eindhoven in the Netherlands. And in the last year of my study, so mainly I was focusing all that time on food and food research. And yeah, I did, like I went to volunteer for NOMA for a bit, and then I got really a lot into foraging. So I went to, and also because of my Russian background, and in Russia, foraging is like really something that people often engage in, just like going to the forest and collecting food. And so after like being so inspired by the community around Noma, I went foraging in the Netherlands, and but I was quite unexperienced, and I almost poisoned myself, which was, yeah, like a frightening experience, because I had thought, so I go out into nature and, you know, like nature has everything we need, and why do we have all this like food chemistry and everything? And so I ate some poison hemlock, which I thought is wild carrot, super, well, it happens. But after that, I realized that in nature, things are actually trying to kill you when you eat them. So there are limits to that. So there are limits to our relationship, with, like to our maybe also romantic relationship with nature. And then I started, like it kind of all came cascading down to me. And I started being more skeptical about our relation, our wish to have everything natural. And if there is, I wanted to know if there is not, like if we cannot turn it around and just get the best of both worlds, like why are we so, like why do I have to see all these labelings, so like contains no artificial flavoring, contains nothing artificial, even though we live in a completely artificial environment and we wouldn't be able to live outside of it. So, and like, and in supermarkets, nothing tries to kill you, which is amazing. So this is, I think, because I had this experience of like trying to become part of the environment and being rejected in this interesting way, I think I had the urge to reframe the way, like to reframe edibility in a way for me. And trying to make sense of it and how can this be translated to something related to community. I wonder about us as a society, how we formulate our relationship with nature and with synthetic, like the role of the synthetic. Yeah, if that answers the question.
Do you feel like you want to move this project in another direction, or is this sort of, this is the project and then you'd be moving on to another one? I'm curious about how, like, based off of what you were just saying, like maybe making it more obvious to people, like how to think more about flavor extractions and this thing from natural grown in ground to where we have it now, this like molecular art strawberry object.
So I think if I had to do the project again, then I would probably do it differently. But I think, I mean, I made it, so it's there, but this is not where like 24 molecules of strawberry is not where flavor ends, right? So I think, so what really fascinates me is, for example, that we can make, or like in biotechnology, it's possible to make vanillin out of wood or cow poop or something like that. So I think this is very interesting for me to approach more in future, like how we can make edible things from non-edible things, and like this, just blurring the borders of edibility through flavor, and also, yeah, just asking questions about what edibility means, because also it relates back to Corinne's work about, yeah, why, like, some people think this is edible and others don't, and like what, like there is no complete answer to that, even though we think it is, because it, yeah.
Can I just add to that? I was thinking the same thing, Alexandra. Like, I love, Marissa, that you paired us together, because we're both still storytelling it with what is edible and what are stories about it. It's just so fascinating. And Alexandra, you're coming more from the angle of obviously from synthetic and what we eat without thinking about, and how these synthetic flavors are defining our flavor profiles, like how we may not even be aware how our sense of taste and flavor, our vocabulary flavors have been defined by these select few companies in New Jersey that are doing all the flavoring. And then from my end, it's like, well, there's different tastes out there. And we eat seaweed growing up, we eat little crayfish from the creek, we'd eat algae, and my grandparents would tell me how they would eat grubs, wood grubs. So they had to supplement their diet on the plantation. They were, my grandfather's from a sugarcane plantation on Maui. And I remember interviewing a bunch of their friends at a hairdresser's once. This is like over a decade ago. And they're just all a bunch of people sitting in the hairdresser chairs, and actually it was a barber shop in Maui. And they were talking about their childhood with such affection for the foods. But part of that was they each had a little bag of it was a Bull Durham bag because everyone chewed tobacco then. So they each got one from the parents, and they would put salt, some Japanese spices in it, and they'd carry it with them in their pocket. Because when they saw workers going out to knock down a wooden fence on the plantation or something, they would go running, and they had their digging sticks. So they had special sticks that they kept with them as well. So they all had their little maker toolkit or scavenging toolkit. and they would dig in the wood and dig out wood grubs. And they would take those and they would dunk it into their little boulder and bag of spices, and they would eat them. And sometimes it would like make little fires that they'd eat them and fry them, but a lot of times they just eat them. And it was something they had affectionate memory for. That's part of their flavor memory that like I definitely don't have. But I just thought that was so interesting. Like they were supplementing their diet, which they needed to do.
Yeah,
But that flavor,
I
right?
just want to say, this is so great. I just want to say it is 1:45 now.
Oh, sorry.
No, don't be.
End up eating grubs.
I love the grub talk. And there's really great talk about foraging happening in the chat. And I feel like we're all into this and maybe can continue the conversation through the Slack channel. I just want to thank everyone for attending. And there will be another tour tomorrow at 8 a.m. And Carolina will be the tour guide for that.